My name is Evelyn Alvarez-Mendoza, and I am a master's student in agriculture with a specialization in crop science at Cal Poly San Luis Obispo. My research is investigating fungicide resistance and botrytis cinerea populations on wine grapes in the central coast of California. When managing botrytis sponge rot in vineyards, the application of synthetic fungicides remains a primary control method utilized by growers. However, repeatedly using fungicides that have the same mode of action leads to fungicide resistance in the pathogen population over time. Resistance accumulation is a widespread issue because it reduces the effectiveness of fungicides and therefore jeopardizes disease management. For my research study, I'll be collecting grape cluster samples from multiple vineyards located throughout the Central Coast region of California. These will include clusters that show active botrytis growth, as well as asymptomatic clusters which will be incubated to promote sporulation. Eventually, I will obtain pure botrytis cinerea isolates from these samples. The isolates will be screened for resistance against 6 different mode of action fungicides, which are all commonly used to control botrytis in the Central Coast. To do this, I will be amending artificial media with these fungicides at 2 discriminatory concentrations. After a certain incubation period, I will then measure the growth of the isolates to determine their level of sensitivity or resistance. My overall research goal is to assist wine grape growers in the Central Coast in the efficient and economical management of their vineyards by relaying information that will help modify fungicide programs in order to achieve successful botrytis management. I would like to sincerely thank the American Wine Society Educational Foundation, and in particular the North Alabama chapter, for supporting me and my research with this valuable scholarship. Thank you so much.
Hi, my name is Sarah Bogenrief, and I am currently a graduate student at North Dakota State University, pursuing my master's degree in plant science. Specifically, my research is in viticulture, and my thesis involves evaluating new and veteran cold-hardy wine grapes used within our region, using differential thermal analysis, otherwise known as DTA, and assessing phenological changes throughout the growing season. In North Dakota, routinely many vines fall victim to winter injury, so being able to monitor the vines throughout the year and not just throughout the growing season is crucial in understanding the health of our vines. My first experience in winemaking was at a very young age, helping a family friend prune their vineyard and bottle their wine. Since then, I have had the opportunity to work with small batch winemaking or microvinifications using experimental crosses at North Dakota State University, while also assisting with large batches at a local winery. Although this work is very tedious, it is also very rewarding, and no hardship goes without many lessons learned. My favorite part of working within the grape and wine industry is connecting with the growers and winemakers themselves. Talking about their vines and their wines is almost like talking to them about their children. My passion comes partly from all those hardworking people in North Dakota looking to advance our small and upcoming wine industry. Helping people using our research is one of the most rewarding aspects of this work. This coming semester, I will continue working on completing my thesis to meet graduation requirements, and our team will continue our work in assessing cold hardiness of both new and veteran grape cultivars. Wine-wise, we will continue to make microvinifications from our breeding vineyard and continue experimenting with local cold hardy fruit wines like Haskap and Juneberry.
Hi, my name is Hannah Charnock, and I'm heading into the 3rd year of my PhD in biological sciences at Brock University with a focus on enology and viticulture. I'm very grateful to have been selected as one of the recipients of the 2021 AWSEF scholarships, and specifically thank you to the Cleveland, Ohio chapter for your generous support. My research is focused on understanding different chemical reactions that drive flavor formation during sparkling wine production and aging. And specifically, I'm interested in something called the Maillard reaction, which has been well understood in different heated food and beverage systems. However, some of these products have been identified in aged sparkling wine, where they contribute desirable roasted, nutty, and caramel aromas. Within this research, we're also looking at how individual factors during the winemaking process might influence the potential to produce Maillard reaction products. Some of these include different sugar types that might be used during the winemaking process, or even different metal compositions that can be derived from the vineyard or the winery itself. This research is really exciting to me because of the potential contribution to the wine industry as a whole, looking at how we can potentially improve sparkling wine flavor as well as sustainability by making more flavorful products and optimizing these Maillard reaction aromas during sparkling wine production and aging. I'm also really excited about this research because it ties together the winemaking world with the wine research world, and it allows me to make sure that our findings can be communicated in a meaningful way and that our research ideas also can have an impact on the industry. And again, thank you very much to the AWSEF and the Cleveland, Ohio chapter for your generous support during my education.
Hi, I'm Bailey Hallwachs. I am a PhD student at Washington State University. I am passionate about the wine industry because grapes are a booming industry to the United States economy. And secondly, grapes are historically one of the first domesticated fruit crops humans have produced. My specific research is on grapevine red blotch virus and how it affects grapevine physiology. Grapevine red blotch virus is a threat to the sustainability of vineyards. Grapevine red blotch virus starts to develop at a time when berries are developing their color and leads to the impairment of proper berry development. So grapevine red blotch virus develops a red blotchy pattern and red veins on the leaf, which disables the movement of sugar and phenolics to the berry, leading to price penalties for growers. My specific research will focus on how the cells and the subcellular organelles are responding to viral infection in the grapevine cells. And to do this, I'm using electron microscopy to discover those pathways and how this relates to the entire grapevine physiology throughout a growing season. I want to thank the Smoky Mountain, Tennessee chapter of the American Wine Society for selecting me as a scholarship recipient for 2021. Thank you so very much, and I hope you have a wonderful rest of your 2021.
I'm a master's student at UC Davis researching how overhead shade plastics work to reduce negative impacts on grape and wine quality under climate change conditions. With temperatures rising and droughts becoming more frequent in California's grape-growing regions, growers are looking for innovative solutions to reduce sunburn and quality degradation in the vineyard. At the UC Davis Oakville Experimental Station in Napa Valley, I am experimenting with overhead shade plastics that are designed to filter and reduce the amount of sunlight reaching the vines. The goal of this research is to determine the appropriate light and temperature conditions under the shade plastics to maintain crop yield and high-quality fruit production desired for winemaking. To determine this, we look at changes to both vine performance and fruit development throughout the season, as well as how these changes in fruit quality directly transfer into wine during the fermentation process. What I love about my role in the wine industry is that I get to use creativity and ingenuity to directly improve crop and wine production, and I get to share my discoveries directly with growers who are interested in adopting new technologies to combat current climatic challenges. The unique aspect of the wine industry which I love most is the combination of art, culture, and modern science created by the tight cooperation and companionship between academia, grape growers, and winemakers. While each group excels at their specialty, we are all working towards the common goal of producing great wine. With that being said, I'm honored to be a recipient of the 2021 American Wine Society Educational Fund Scholarships and would like to especially thank the Thomas Jefferson Kentucky chapter for their support and generosity.
Hello, my name is McGonigal. I'm a PhD student studying under Dr. Louise Nelson at the University of British Columbia, Okanagan campus. My research focuses on crown gall disease of grapevine, an economically devastating disease that tends to be most severe in cold climate grape growing regions. In grapevine, crown gall is caused by the bacterial pathogen Alvarez-Mendoza.
The common visible sign of crown gall is the development of tumors along the trunk. Symptoms of crown gall include reduced crop quality, decreased plant performance, root necrosis, and even partial or complete vine death. The main issue with this disease is that there's no reliable management strategy to turn to. In my project, I am testing compost application as a potential strategy for managing crown gall in the field, as well as testing how other microbes may be able to prevent crown gall in greenhouse trials. I'm also collecting tumor tissue off diseased vines from across the Okanagan Valley, isolating A. vitis, and then completing phylogenetic analysis of the strains to estimate their genetic relatedness. This analysis of local strains is important for improving our understanding of the pattern and distribution of this pathogen.
My research is important to the North American wine industry because crown gall can put a severe economic strain on affected vineyards. My goals for this project are to identify sustainable management strategies that grape growers can implement in their vineyards in order to combat crown gall. Ideally, this will result in improved wine quality, protection against economic loss, and the preservation of our beautiful North American vineyards.
I would like to thank the Northeast Pennsylvania chapter of the American Wine Society Educational Foundation for this scholarship, as well as the opportunity to share the research I am so passionate about.
Hi, my name is Demi Perry, and I'm a PhD candidate at Cornell University in Ithaca, New York. My research focuses on how to take Concord juice and make it more suitable for premium red wine production to not only benefit grape growers, but also winemakers here in North America.
When I first ventured out on this journey, there were two promising approaches to try to transform Concord into something palatable, delicious even. The first was flash détente. Flash détente utilizes heat and vacuum expansion to volatilize aromas present in the skin of the berries. It is often used on the West Coast to reduce methoxypyrazines and underripe grapes. It is also thought to create a more colorful juice. Nanofiltration resin treatment, or NFR for short, is a two-step approach that engages a spiral wound membrane to retain color and large macromolecules in juice while allowing small molecules, like the ones responsible for aroma, to pass through. What passes through the membrane, the permeate, is then channeled into a resin canister that is semi-selective based on a molecule's chemistry. This system allows us to start with grape juice that smells and tastes like Welch's grape juice and end with a juice that is just as colorful, but without the distinct purple grapey flavor, for a more neutral starting juice. In the upcoming year, I hope to classify the extent of grapey removal by comparing data collected from quantitative lab analysis using GCMS and UV-Vis to sensory data. In other words, do wine drinkers find my wine any less grapey than a control grape juice? This sensory data, along with data amassed in the lab, will allow us to make predictions of the success of this processing technique and incorporation into the market. This work would not be possible without funding support from New York Ag and Markets, Welch's, and last but not least, from the support of the American Wine Society Educational Foundation. Thank you.